Welcome back to my channel, and today in this video we are going to study about physical basis of inheritance. So as you all know that in our body or we are made up of cells. Cell contain nucleus. Inside the nucleus we have chromosomes. Chromosomes contain chromatin. What is chromatin? DNA plus protein, specifically histone. These are DNA wound around histone protein. So chromatin. Now this chromatin contains DNA, and in this DNA we have genes. This we all know. Now our cell divides due to mitosis, cell number increases. Due to meiosis, gametes formation take place. Now our cell cycle consists of these phases, that is G1 phase, S phase, G2 phase and M phase, which is known as mitosis phase. So the cycle starts with G1 phase. What we have in G1 phase is 46 chromosomes. 23 chromosomes from father side, 23 chromosomes from mother side. So total our each cell contain this 46 chromosomes and 46 chromatids also. Chromatids means this is known as chromatid. One chromosome. So 46 chromatids, and the center part, this is known as centromere. So 46 centromere. Why I am specifically pointing centromere and chromatids also, we will see now. So because we are diploid organism, we have 2n set of chromosomes. So 2n means 1n from mother, 1n from father. So total 23 such pairs. This blue 123, this yellow 123, a total of 46 chromosomes our each cell contains. Now what happens in S phase? S phase means synthesis phase. Here DNA replicates. Correct. Our chromosome replicates. Now this father 23, it will also replicate. This mother 23 will also replicate. So now how many chromatids? Earlier it was 46 chromatids. Why? Because this blue one and this yellow one all single present. So 46 chromosome, 46 chromatids. But after replication, now we are having two chromatids in each chromosome. Correct? So total number of chromosome, we still say it is a 46 chromosome. When it is single, then also we are saying it is 46 chromosomes. When now after replication, still we are saying it is 46 chromosomes. Why? Because we count the number of chromosomes on the basis of centromere number. So here also this one chromosome is having only one centromere. Here after replication they are still connected with one centromere. So the number of centromere remains same. Hence the number of chromosome also we count it as a same. When it is single, still 46 chromosome. After replication, still 46 chromosome. But the number of chromatids are more. Here in G1 phase 46 chromatids. Here each chromosome is consists of 2 chromatids, therefore total of 92 chromatids. So this set blue one from father, this set yellow one from mother. So from father and mother we get 46 chromosome, this single, and then in S phase we replicate it, make it double. Now these two set 2 chromatids, 1 chromosome of father and 2 chromatid or 1 chromosome of mother, these we call it as a homologous chromosome. Because we are getting 23 from father, 23 from mother. So here also chromosome number 1, here also chromosome number 1. After it get duplicates, it is 1 plus 1, it is now 1 plus 1, but still we call it as a 46 chromosome. Now this 1 plus 1 and this 1 plus 1, these are known as homologous chromosomes. Clear? Then again in M phase, the cell will divide and again in each cell will get 46 chromosome, 46 chromatids. So in somatic cell, we can see mitosis. In germinal cell, we see meiosis. Now, in animals and in plants, the process is male and female, both are having 2n chromosome. They'll form gametes, undergo meiosis, and each gamete will have one n set. With 2n, we can get 4 types of gametes because as you remember, after duplication, when separation happened, so we are having total of 4 chromosomes. So 4 gametes we can form. Now, In case of male, all remain alive, and when one end of male fertilize with one end of female, the rest three gametes, they get degenerated. So this, suppose this N and this N, they'll together form 2N of zygote, and then it will undergo mitosis, and during gamete formation, meiosis. This happens in animal. Now what happens in plant is it's the same. The male and female is having 2N chromosome. They'll form gametes. One N from father, one N from mother. They'll combine. Rest will get degenerate. So they'll undergo mitosis first, and then they form sperm and egg. After they form sperm and egg, then it combines to get 2N zygote, and then again it undergo mitosis to increase the cell number. So this is little bit a difference between animal and plant. In animal, it is like they form 2N zygote and then undergo mitosis. In plant, what is happening? They'll undergo mitosis first. Before combining these ends, they separately undergo mitosis. Then from there, some N will behave as a sperm, will convert into sperm, and some N will be egg. Then they will combine to form 2N zygote. So now we will understand how law of segregation and law of independent assortment works in meiosis. Why meiosis? Because we are studying genetics. That means we are studying genes that are transferred from one generation to the next generation. So when we are talking about transferring of gene from one generation to the another generation, this transfer happens via gametes, and gametes are formed due to meiosis. So now we will understand how law of segregation and law of independent assortment work in chromosome during meiosis. So this is our interphase state where we are having one set of chromosome from father and one set of chromosome from mother. So we are having 46 chromosomes and 46 chromatids as well. So here we are talking with the gene capital T and small t. One from father side, one from mother side. Now in interphase only the DNA will replicate or the chromosome will replicate. Now when these chromosomes will enter in prophase 1, why prophase 1? Because we are talking with respect to meiosis. Meiosis is divided into two phases, meiosis 1 and meiosis 2. So the stages of meiosis 1 are labeled as prophase 1, metaphase 1, anaphase 1, and similarly for meiosis 2. So when the chromosome reach to prophase 1, now it is duplicated. Still we call it as a 46 chromosome, but the chromatids here are 92. So now it is TT and small t, small t. Now they will segregate in the anaphase 1. There is no crossing over happen for this process. So when there is no crossing over, that means only two types of gametes will form, the capital T1 and the small t1. So when these two will get separated, they will form the same type of gamete, capital T and capital T. When these two get separated, they will form the same type of gamete, small t and small t. So when there is no crossing over, the segregation of the alleles happen at anaphase 1. So in anaphase 1 only they are now segregated, they are now separate. By anaphase 2 they will separate in 4 cells. They will get separated in 4 cells. So we are having only two types of gametes, capital T type and the small t type, because this is the condition when there is no crossing over. So when there is no crossing over, the segregation or the law of segregation works at the anaphase 1. Now what happens when there is a crossing over? So this is our interphase state. One chromosome from father, again one chromosome from mother. In the interphase state only, it will get duplicated, double during S phase, synthesis phase of interphase. In prophase 1, if you remember, during meiosis, the prophase 1 is the state where the crossing over takes place. So here you can see this small t now is on the blue chromosome. This capital T, pink one, is now on the yellow chromosome. So that means these two get, the crossing over happen between these two chromosomes. Now this is the anaphase 1. In anaphase 1, this capital R, capital T, capital R and small t, these are still linked together with one centromere. Similarly, small r capital T and small r small t, they are linked still in anaphase 1 through the centromere. Correct? During anaphase 2, they are all now separated or segregated. So the law of segregation, when crossing over happen, that time the law of segregation work during anaphase 2. When there is no crossing over, the law of segregation works during anaphase 1 because in the absence of crossing over, only two types of gametes are formed. So they are segregated. Capital T is completely different from the small t. But here the capital T and the small t are still linked together through one centromere. Though they are present in different chromosomes, but these two chromosomes are linked with one centromere. So this centromere separation happens in anaphase 2. That's when the capital T here and the small t here get segregated. So when there is a crossing over, the law of segregation works at anaphase 2. When there is no crossing over, the law of segregation works at anaphase 1. So this is the condition when crossing over occurs. So now what we understood, that law of segregation is the universal law. Whether or not the crossing over occur, the law of segregation will always happen. It will happen whether or not crossing over occurs. If there is no crossing over, then segregation occur at anaphase 1. If there is crossing over, then segregation occur at anaphase 2. Now come to the law of independent assortment. Here how many types of gametes are formed? 4. Capital R, capital T, capital R, small t, small r, capital T, small r, small t. So 4 different types of gametes are formed, and this happens when the genes are not linked. R is not linked with T, hence with capital R, small t can also present. So this happened because the crossing over happened. Crossing over happened proves that the genes are not linked together. That means the genes can assort independently. Any gene can present with any other gene. So that is known as law of independent assortment. But there are few criteria that are required to be fulfilled for law of independent assortment to be happen, that we already studied. So we won't discuss it here. So the formation of 4 type of gametes prove law of independent assortment. The maximum variation in individual is due to law of independent assortment that we already studied. Further variations are created due to crossing overs. So law of independent assortment alone is responsible for the maximum variations. Furthermore, more variations are created due to crossing over. So this is only known as physical or chromosomal basis of inheritance. Now one question from my end for you all. What is the difference between law of segregation and law of independent assortment? Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.